So we were here in last video. So the some content of this slide is hidden because this video where I am is overlapping with some of the content. So now if you look this right hand side table, which actually has sex chromosome complement, then haploid sets of autosomes, X to A ratio, and sexual phenotype. Now, if there are two X to A, then the ratio between X and A will be 1. So the sexual phenotype will be female. If it is XYAA, then the ratio will be 0.5, and we have just discussed that then the sexual phenotype will be male. If it is X0 AA, then again the ratio is 0.5, and what is important to note it is the phenotype is male. So what is the difference is that in case of the humans we discussed that the presence of Y chromosome basically determines the sex, whereas if you will see that there in the like if there is no Y, still the sexual phenotype is male because the Y chromosome in Drosophila do not determines the sex sexual phenotype, whereas it only determines whether the male will be fertile or not. So if the ratio is X, so now it is XXYAA, the ratio is 1 female, triple XAA ratio is 1.5. The sexual phenotype will be meta female. Now it is the work for you to look for the features of meta female. Now if it is triple XYAA, it's understood the ratio is 1.5, it will be meta female. XX triple A, it is 0.67. Now here it is the sexual phenotype will be intersex, which is both, it is both male and female. This X not triple A, it is 0.33, it is meta male, whereas triple A is 4x triple A 1.3, it is meta female. Now coming to the molecular mechanism of sex discrimination in Drosophila, when I talk about the molecular mechanism, I will be specifically talking about the DNA, RNA, and proteins. Now we know that there are some kind of signaling mechanism always going inside our cellular system, which controls the overall metabolic pathways. Similarly, here also how we will discuss about how the molecules are going to decide the fate of the embryo, whether it will be male or female. Now, to start with, we will be discussing about the 6 steps. First step is the first very much important is the establishment of X to A ratio, which is and now the second is conversion of XX to A ratio into the molecular signals. Now the differential activation of XXL gene, which is very important. XXL gene here means the sex lethal gene, which will be discussing about this gene in upcoming slides. Now the differential regulation of transformer gene, which are also called as TRA genes. I repeat, it is TRA gene. Now the action of double sex gene, which is DSX, the switch gene of the sex determination, which is very important, and it is at the last where it determines what will be the sex of the embryo. Now what is and the production of male or female. Now, first to start with the establishment of X to A ratio. Establishment of X to A ratio involves interaction between the proteins which are encoded by the X chromosome and the protein by the autosomal chromosome. Now, as we just discussed that the X to A ratio will going to determine the fate of the embryo. So X was a numerator and the A was denominator. Basically, it's a tug of war between X and A, the protein which is coded by the X gene and the A gene. If the proteins which is coded by the X if are more, then the numerator is more and the embryo will develop to female, whereas if the denominator, that is the proteins which are encoded by the autosomal genes, are more, then the embryo will be male. So X-linked genes are also called numerator genes, which are examples is A, cis, B, cis, C, and RAN. So this means X-linked, I guess you are very much aware of it, that the genes which are present on X chromosome, they are called as X-linked. When I say autosomal genes are called, here in this case, the autosomal genes are also called as denominator genes, example DPN and EMC. We will look into each and every gene. We will talk about the pathway in upcoming slides. The denominator gene encodes the denominator protein, which antagonize the numerator protein. When I say the word antagonize, it means denominator proteins are going to function opposite of what numerator is going to do. So that is the word antagonize means. So in XX embryo, when the denominator protein is more than is less than numerator protein, the embryo will follow the female pathway, and in XY, the denominator proteins it will be more than numerator protein, and now if you look into this chart, starting from the from left, uh, if I can take some pen or anything. So this, I'm talking about this region. The, now if you look into this chart, starting from the from left, uh, if I can take some pen or anything. So this, I'm talking about this region. The numerator genes will code for the numerator protein. Similarly, the denominator genes which are present on autosomal region. So or on autosomal chromosomes, and these are present on X chromosomes. Now they are coding for numerator protein. They are coding for denominator protein. What we can see that the numerator and denominator, they are binding to each other, whereas what we can see, the numerator proteins are more, so they are free. So the free numerator protein can lead to the activation of the sex lethal gene, and this activation will lead to formation of RNA. Now coming to this, look into this here. I'm talking about this. So they have X and Y chromosome, and similarly these are the autosomal. These are these are autosomal regions. So they are coding for the numerator proteins. They are coding for denominator proteins. They are binding to each other. In this case, the six lethal gene is what you can see, that the denominator proteins are more. So that this is what exactly we discussed. And now if you look, that six lethal genes will not going to be active, they will be inactive. This is hidden. You can look into the PPT alone. So they are hidden. So the six lethal gene will be inactive in this case. Now again I can select this. So now how the ratio is going to give a molecular signal. How the ratio is going to give the molecular signal. So the XXL is the master regulator of the sex determination pathway. So what happens that when their Drosophila embryo has a chromosome which is XX, in XX Drosophila, the XX is active during the first 2 hours after the fertilization. Now, XXL gene transcribe a distinct embryonic type mRNA from promoter PE. So the promoter PE means the early promoter. So the XXL gene, we know that the genes are composed of promoter. The gene consists of promoter. I I I, I hope that you are aware of term promoter when you have read the lack uh, about the lack of iron in your class 12. You are very much aware that promoter is a region where the RNA polymerase binds, and after which the transcription starts. So in case of the XXL gene, in case of the sex lethal gene, there are two promoters. I repeat, there are two promoters. First promoter is the early promoter, which will express early. That's why the name is PE, early promoter, and it will express itself just and it will during the first 2 hours after fertilization. The early XXL mRNA. Now this I'm talking about this. The early XXL mRNA, which is encoded by this promoter are processed and translated into the XXL protein. Now, what I'm repeating it again. In case of XX Drosophila, what will happen? That the XXL will be active during the first 2 hours with the help of this early promoter, and this will we know that the gene will encode for the mRNA, and mRNA will encode for protein. And I repeat, and I will tell you again and again that not all genes code for protein. We know that there are some genes which don't code for proteins, such as tRNA genes or rRNA genes. Whereas if we see again, so what's happening? This XXL mRNA is going to be processed into XXL protein. This protein, after few division, now after few division, the transcription starts from the PM. PM is the maintenance promoter. PM means M means here maintenance promoter instead of PE. So the transcription from the maintenance promoter is also initiated in XY embryos. Whereas if in case of XY there was no PE, there was no PE promoter activity. So there was no early XXL gene, the but in case of XY embryo will only have PM, that is maintenance promoter mediated XXL gene expression. So I am repeating it again. So to make you clear is that when there is XX, there is XX, there is early expression of XXL gene with the help of PE promoter, that is early. Now the early XXL mRNA is going to form the XXL protein. Later on, after few division, transcription starts from PM promoter, which is maintenance promoter, instead of P in case of XX Drosophila. Whereas in case of XY embryo, now if XXL protein is already available from the early translation, now who was helping in the early translation? You know this, that is PE. And this was only present in, sorry for this very bad writing. So in XXL protein is already available from early translation. The XXL pre-mRNA is spliced to form functional female specific message. This encodes for functional proteins. So the early expression is coding for a protein called which is 314 amino acid long. In wild-type Drosophila with one X chromosome and two sets of autosome, now here what we can see is that here we have two sets of autosomes and one X chromosome. So we know that this is going to be male. So XXL, sex lethal gene is transcribed from late promoter. That is PM. In the absence of the early XXL protein, we know that the early XXL protein is not present in case of when there is only single X, or when there is a ratio of 0.5. So when there is no early XXL protein, that RNA splicing does not exclude the male specific exon in mRNA. We'll look into this in next slide. So what I actually am trying to tell you is that if there is XX chromosome, if there is XX chromosome and the ratio is 1, then what will happen that the early promoter is going to form XXL protein, and later on whereas in comparison where only the 1X and the 2 autosomes are present, in this case the early expression of XXL gene is not there. So when there is no early expression of XXL gene, then there is no XXL protein. When there is no XXL protein, this XXL protein is not going because this XXL protein which is formed early because of PE promoter, is actually helps in the splicing of the mRNA. I repeat, splicing of the mRNA. Splicing means chopping. So splicing of the mRNA of XXL gene expression, XXL gene made mRNA which is coded by. See, this is first I will talk about XX embryo. XX embryo codes for 1, 2, 3, 4, 5, 6, 7, 8. 8 exons are there. 8 exons are there. So this dashed box are exons. These lines are introns. So we have 1, 2, 3, 4, 5, 6, 7, 8. We have 8 exons. If you see this, the number should be 1, 2, 3, 4, 5, 6, 7, 8. But what it is? 1, 2, 4, 5, 6, 7, 8. So 3 is out. It means that the tree has been spliced. So tree has been chopped. This tree has been chopped by the early X X SXL protein, which was which was coded by PE. Now this XXL protein, which was coded by PE, is actually cleaving off this third exon, and it will form a XXL protein. It will also form the XXL protein, but this is from PM promoter. So we discussed that the XXL gene has two promoters, PE PE and PM. This PE codes for XXL, helps in coding of early XXL protein. This early XXL protein will splice this third exon of this mRNA, which is coded by with the help of PM, and it will only have 1, 2, 4, 5, and it will code for the functional XXL protein. If you see XY, so here we have 1, 2, 3, 4, 5, 6, 7, 8 exons. If you see this, the mRNA is having the third exon, and what is wrong with third exon is just that this third exon have termination signal. This termination signal actually do not form the functional polypeptide, which was actually formed in case of XX embryo, where the third exon was chopped off. Why not? Why not this third exon was chopped? Because in case of XY embryo, we learned that the PE mediated expression was not there. When PE mediated expression was not there, then XXL protein early will not be there, and the protein which is coded by PM, the XXL which is coded by PM, will have this third exon which will not be spliced off, and this third exon has what? The termination signal, and this will form the non-functional polypeptide. So if we go by this, in male, the mRNA is spliced in manner that it yields 8 exons. So here we have 8 exons, and the termination. So what I will repeat that the XXL will lead to the splicing of this third exon. Okay, in and if this early promoter mediated XXL protein is there, it will lead to splicing of this third exon, removal of this third exon, and that will lead to the functional XXL protein. But in case of XY embryo, PE expression, PE XXL mediated expression is not there. It will not lead to formation of early XXL protein, which is not going. If it is not there, then there is no removal of third exon, which harbors what? So termination codon, and there will be non-functional polypeptide. In female, RNA processing yields only 7 exons. So 1, 2, 3, 4, 5, 6, 7. And the male specific exon 3 is spliced. It is spliced out as large intron. I guess this part is clear. I am ending it here. I hope that uh, you people will be able to get it. So I will be making a separate video, and will continue.